And as a pharmaceutical physician with a translational medicine background, I know firsthand how critical and challenging it is to navigate the path from the preclinical research towards the clinic. And this session is all about those pivotal decisions and strategies that determine whether a program reaches patients or stalls at the bench, or as Kinga and some others called it, the valley of the death, or I like to call it lost in translation. And before we dive into the conversation, I'd like to ask each of the panel members to introduce your background and your role, and what are you bringing to the panel today?
So my name is Marin Mladinic. I'm hired by PPD. By background, I'm a board-certified toxicologist. I have almost 18 years of tox experience encompassing academia, pharma industry, and now CRO side. So I basically do everything from lead candidate selection to first in human and later stages, so up to NDA, BLA. So that's my experience.
So my name's Chris Floyd. So I'm also a pharmaceutical physician as well as Judit. My background is I spent around 15 years as a clinical academic working in clinical pharmacology, general medicine and drug development more broadly. I then recently spent 4 years at GSK within the early space, taking a number of molecules into first time in human. At the moment, I'm an executive medical director at tranScrip, which is a pharmaceutical consultancy. And there I advise early phase clients on essentially how to get into the clinic. So either as a CMO role or more as a physician to lead their project, depending on the size of the organisation.
Yeah, so hello again. My name is David Cook. I'm the CEO of Quodria. Prior to that, I worked for AstraZeneca for a thick end of 20 years in a variety of roles. I actually helped set up AstraZeneca's Systems Biology Group in 2000. I was saying earlier that was way too early. That was a complete failure. But anyway, different story. I worked in both the efficacy and safety of medicines side of things, and was also heavily involved in a big review of AstraZeneca's portfolio, which became the 5Rs paper, which got published about 10 years ago in Nature, which I was the first author. Since leaving AZ, I was CSO at Blueberry Therapeutics, and now I'm the CEO of Quodria.
Hi, my name is Joe Stanta. My background, I've been for the last nearly 15 years in leadership roles in CRO and the pharmaceutical area, mainly in the region of bioanalysis, biomarker and CDX development, and also very much on the interface of translating preclinical to early clinical and accelerated clinical development. Yeah, great to be part of this panel.
Hi everyone, my name is Debra, one of the co-founders of DotPlot along with Shefali. I have a background in product design and innovation design engineering. Did some work with the German Aerospace Center developing interactions for people with upper limb prosthetics. And now, yeah, working on DotPlot, looking to bring breast health monitoring to the home space.
Thank you very much. So to kick things off, let's ground ourselves in the earlier stages of drug discovery. What are the most critical decisions you need to make in the earlier stages of drug discovery that can either de-risk or derail clinic readiness?
Can I take that one?
Yes, please.
So I would highly urge all of the companies to think about the species selection. So basically, the animal model you're going to be used to get your product in. So analyze and do it very early. So if you use the wrong model, you're actually throwing money and animals into the wind. So very early on, you should pick the right species. Of course, different modalities have different rationales how to pick them. But for small molecules, start with metabolite identification. For large molecules, it would be more with pharmacological relevance. So that's a key thing you should have to be considering once you have your candidate. Where do we go next and into which species? Are we able to do it in these animals? If not, you are required to show it to regulatory bodies. You cannot use any species, so you might go down the route for in silico, in vitro, something else.
And Chris, I know what are you going to say, because we are coming from a very similar space. So pretend, yeah, let's just...
I'm going to surprise you then.
Okay, say something else then.
So I would say, I completely agree with your point on tox species, but I think all of the decisions you make early on can have a massive impact down the line. And probably the most important thing in my mind is defining your target product profile or your TMP. You know, if you decide that you want to do a drug for liver disease, you need to define what level of hepatotoxicity you are acceptable at the start. Because if you don't know what route you're going to take, you can suddenly end up with a load of candidates down the line which are not suitable for the disease you're after. And to give an example of how these kind of issues can cascade, if I think about CMC, and Dave, you gave a great example, if you don't make enough CMC for your preclinical toxicology, you can't go high enough. If you can't go high enough in your preclinical tox, you can't go high enough in your phase 1. You take a too low dose into your phase 2, and suddenly the best molecule in the world is failing in phase 2, all because of a decision to make 800 grams, not 1,000 grams, taken 4 years ago. There's that cascade that I think is really challenging.
Yeah, I would agree with you. I think the TPP is the single most important thing you start with. Understand what is it you're going to make, who's it for, why should they take it, how are you going to know it works? And then everything else is about working back from there through the different processes to understand all of those implications, as you say, and get back to the lab. I mean, certainly in the 5 Rs, we very much focused on then abstracting key information, writing it down. I know that sounds stupid, but understanding your assumptions with respect to what's an assumption, what's a fact, what's real, where are the questions. And I would say the general mode of action we always take is don't be scared to break your project. Ask the difficult question. You should be a bit nervous about it. You know, what's the answer going to be? Because if you fail to break it, it will move on to the next and be stronger for it.
Yeah, I think really good point. Maybe what I can add to this as well is that, you know, especially with biomarkers, focus on showing an early readout of efficacy as well. If you implement that very early on, it gives you a chance, while you might not need the data straight away, but it gives you an early chance to really start to validate that biomarker, validate in terms of not as an analytical way, but in terms of an actual usability in the disease that you're after. And that can really, and there's so many examples of that in the last 2 years, where you know, biotech companies have been bought out for like a fantastic amount of money, great purchases, because in healthy volunteers they could already show efficacy. Even before moving into phase 2, they could show the drug absolutely works and it hits that biomarker very nicely. And that's something that, again, can be sort of, you know, preloaded with regards to having the assay ready when you need it, but also start early with the development. Make sure your reagents are there, your platforms are there, and you get time to test and try and maybe also reject some of them if you don't need them, but have a think of that very early on. That's where, at the moment, especially in biotech space, where you will run out of money to run a phase 3, most likely, so you need a partner, you need someone. This is where you can show early on the value of the molecule that you have and the value. And this can be the difference between a factor of 10 in the company evaluation if you can show that very convincing data that really, you know, shows the proof very early on.
I think for us in medtech, it's really important to understand the problem that you're solving early on. Understand the problem really well. So with us, we knew that women were having a hard time understanding or differentiating what they were feeling within their breast tissue when they were doing self-checks. So what sort of technology can actually help to address that problem in a way that's simple, understandable, and usable by a lay user. And then, yeah, figuring out what's the simplest way to actually solve the challenge that we've identified with these women. Because with hardware, we learned very early on that building hardware is hard. It's in the name. And so you want to find the simplest way to address that problem. I think it was Marin in an earlier conversation that was saying, with innovation, it's not always about creating something totally novel. Sometimes it's taking something that exists already and adapting that and pushing that into a market in a way that we haven't seen it done before. And so with DotPlot, we were like, yep, this is a big challenge. What's the simplest way that we can solve it? So those were the kind of early questions we were trying to get sorted in the beginning.
Thank you. And my next question would be that what makes a preclinical package truly clinic ready? So are there must-have data points or experiments that you would never skip?
Okay. Should
I know we need to do tox studies.
I think
in a broader...
again, it all comes back to understanding your product, because then you define what bluntly, efficacy first, safety second, and that's no disrespect, because if you haven't got something that you don't believe is going to work,
it doesn't matter.
It doesn't matter.
Yeah.
It can be as safe as houses, but if it doesn't have any efficacy. But you need to understand, again, your product. What does that package look like? And I say, I'm a big believer in you keep trying to break it. You do this experiment, it passed that one, this one. Right, the next experiment is in the clinic, because I've run out of things to kill it in the preclinical space.
And Dave, what I'd add is maybe to pick up on, Johannes, what you were saying in terms of biomarkers. So to get something into the clinic is relatively easy. There are hurdles you need to get over with the regulators, based on the weight of evidence, but, it's nowhere near a tick box, but you can kind of scope out what you need to get there. But to then get from your phase 1 into your more expensive POC phase 2, that I think is the challenge. And that's about building up that translational package on the years that, take you up to running your phase 1. Because phase 1s these days are not just safety, tolerability and PK. We need PD, we need those biomarkers to make that translational link back, probably from healthy participants through to the animal models, the cells, and hopefully back to that observational human data that usually launched our hypothesis.
I totally agree, and I think that's where many companies fall over, or we have seen it in the past, that getting into phase 1, I have a safe molecule, but then what? Then you are going to run into, you have to run a massive phase 2 program because you have no idea where you are on your dose response curve. And that work happens preclinically. And this is when sort of, if you are a cash-strapped biotech, this is where people try to save money. And I'd like to move into the next topic to discuss how do you balance this? How do you balance scientific ambition, budget, and answering all the questions that you want to answer?
I think that's really, one, I would say, if you can actually get into patients early as well, and actually you can get into patients phase 1. It's in a lot of indications, and especially as an expansion arm, it's a good way of thinking about it, early for efficacy. I'm not going to answer your question. I'm going to tell you a little story I heard from an investor. He said that they had a biotech they invested in, and they were super slick, and they got all the way through to their in vivo study. They had brilliant results, and no one believed it. And no one believed it because they hadn't done any of the work up to show exposure, to understand what they were seeing. Yeah. So it was a total Hail Mary experiment, and that was the point, is, if you just put in enough money into a business to just run the slickest experimental plan, it's not necessarily the wisest. So thinking about how you build the story, address the questions, you want to be able to say, this molecule, I know it's there, I know it's exposed, I know it's engaging the target, and now I can see a result and I can interpret it. Yeah,
and it's not doing too many experiments, as Johannes and Dave, you've said, throughout. You don't just. There's always one more experiment you can do, or there's always 10 more experiments you can do. But actually, and I forget who said this, but it's about thinking what do I expect to see from this experiment, and what's the chance of me seeing it. So if you've got an experiment and you're 90% sure of the results, that you're not going to learn much. That's probably not the best investment unless you have to get it from a regulatory point of view. Where the sweet spot is it 50%? It depends on the program. But you just need to really carefully think, I think, how does this experiment improve my probability of success?
Or is it going to change anything I'm planning to do? That's what I usually go back to biomarker scientists when they come with a shopping list. And we are going to talk about biomarkers, that you need to know that what are you going to do with that data? Is that going to change what we are doing? If not, don't spend the money. Yeah.
But it's also not just spend the money. If you generate data you cannot explain, you're opening a Pandora's box of regulatory questions. So mandatory, or what's necessary versus what's nice to have. So trim down nice to haves down to, yes, we need these, versus what can be done extra. And also, do we have the money to run these extra studies? But if your answer is, we do not know what if we get A or B, and you're not required to do it, then I urge you not to do it, because regulators are also scientists. So if you open up with 2-3 things, oh, this looks nice. How about you explain us? And then you're like stuck with another year of development.
Yeah, don't dig up dirt.
Yeah,
That's what I usually...
It's exactly the same in clinical medicine, where you can tick a box and order every single test on someone. But if you order enough tests, statistically, some are going to be outside the normal range. What do you do with that? And it's the same in drug development.
So, shall we talk about the evolving regulatory landscape? And I'm looking at Marin so in April 2025, so not too long ago, the FDA announced plans to phase out animal testing requirements, particularly for monoclonal antibodies, and I think it came up previously already. They are encouraging people to use alternative models like AI simulations, organoids, PBPK modeling. So, have any of you used these alternative models, or what do people think about that? Are we really going to get rid of animal testing with biologics? Is that?
I can say from recent experience seeing meeting minutes from the FDA, although sponsors are starting to suggest that to the FDA, basically following their plan, the FDA is not still keen on saying, no, you don't need. So one sponsor suggested instead of doing a 6-month monkey study, let's trim down, as you said, to 3 months or 1 month. And we will add clinical data from our competitors. And they said, please provide us, and then we will decide. So even if you're following, there's still, I think, a lot of skepsis on that side. On another side, organ on chips, which is a really good idea, but if you use animals for organ on chips, we're substituting animals with animal data on chips. So organ on chips should be done based on human biology, human tissues. This is the goal. So if we remove the animals and uncertainties, then I would say we need those methods, especially like organ on chips, with human samples.
And I think most of us are working on drugs, therapeutics, and I want to bring in Debra because you are working on a device which I know has a different regulatory path. So when did you start thinking about bringing in the regulators, and how did you approach that?
Yeah, so honestly with DotPlot, we started it quite early. So I remember as soon as Chef and I had spent like 3 months on it, working on it during our masters, we definitely wanted to go and find regulatory experts to just give us a heads up about what was coming, what should we expect. So yeah, my advice would be go in early when it comes to regulatory support. At least have an understanding of what it is you're trying to build, what you're intending to create with your drug, tool, device, whatever it is. And then from there, it gives you an understanding of what exactly can you claim with the device, and what are you actually trying to achieve with it, and how long is it going to take for you to actually get regulated. So with DotPlot, we knew from early on that this has to be a medical device. Many people were suggesting, it could be a class 1 device, no? A simple tool, get it easily in and out. And we found that actually, no, it's not that simple. We're not monitoring your sleep. We're not monitoring, just lower risk factors like that. We're looking at breast cancer. And so we can't hop, skip and jump through regulatory, just regulation. So we couldn't do that. So yeah, we got involved with regulatory experts early on in the journey, and that really helped to pave the way for what we can claim and what we are building.
Thank you.
Yeah, maybe.
No, you go on.
Yeah. Maybe one aspect to ask as well. So I think one of the questions was about also the regulatory landscape in general, and I think it's only a draft publication from the FDA where they just would have put an idea out to say like, well, actually, please, especially for monoclonal antibodies, do you consider non-human animal models? It's only a draft consultation, and the timeline of that is sort of 5 years. The thing is that the agency wants to learn a lot more about those models in the data that's been submitted, because as you said, like, you don't want to submit data that you can't interpret or you don't quite know about, and the agency don't see that. If you read the investigator's brochure, that data doesn't go in there. The modeling doesn't go in there because people are afraid to put it in there. So the agency's push is really to say like, well, please come to us and speak to us. We want to learn about it as much. It doesn't mean that you're going to get a drug approved or into the clinic if you just have done an organoid and no animal testing. However, they want to be part of that journey.
Mm-hmm.
And from a landscape point of view, I think this is really great and forward thinking from the FDA. Well, long may it last. Who knows what's going to change next there, you know. But, you know, at least they're spearheading this and putting this out there. EMA or, you know, the MHRA or, you know, Germany as a country, they have not come up with anything like that. They're following along and they're all right with it, but they are not leading the field. They're not saying this is how to do it. And that's from, you know, from a regulatory point of view, especially when you talk about medical devices. In Europe, you have to deal with so many different notified bodies to even get your CE mark and get the advice. And then the UK is different to Ireland and to Germany and Italy and everyone you have to speak to separately, which I think really holds back the continent in terms of, not in terms of the work that you need to do, but in terms of know what to do at what time. So it's relatively straightforward what the FDA is thinking because they're publishing it and you can follow it and you can start the conversation with it. But in the UK, in Europe, it's a lot more fragmented and a lot more difficult. And you gave the presentation, you have to like, it's just a language you have to, if you want to submit to Poland, it has to be in Polish. Germany, it has to be in German. So again, that gives an extra level of burden. And then notified body might not be agree what the outcome was from notified body in France, because there's a different assessor that looked into it. So I think that's definitely an aspect where, and we do see that, where innovative aspects like that get submitted first and pushed first to the FDA because it's a lot more clear-cut in terms of what to expect and which route to follow.
Yeah, I mean, just a couple of notes of caution. I mentioned in our talk, you know, when I think definitely seek regulatory input. So the MHRA, for example, will do a scientific advice meeting for you, really useful. But do think about what you're asking them and what questions you're asking, because the answers to those questions will be documented and they become part of your narrative. I say we made the mistake of asking the FDA too early when we had nothing. So they said, oh, it could be this, this, or this, and these were things that we knew were irrelevant. But actually they lived with the program and still, you know, I think are still living with the program. The other thing I say is don't go regulatory shopping, as someone called it to me once. So regulators, as you've said, they all have slightly different views. So you could go, well, actually, I've asked the MHRA and not quite what I want. I'm going to go and ask B-Farm in Germany. Oh, I'm going to go ask Sukkel in Czech. Still not what I want. Each of those now is creating subtle different versions of problems for you. Yeah. So, yeah, just a few things to be aware of. But I would really encourage you, when you're at a position where you've got your plan, you're thinking about it, as Debra said, seek the regulatory view. They will caveat it with, when you submit your CTA, we will tell you. And actually, that can be a strategy as well. Don't bother with scientific advice, submit a CTA and see what they say.
Yeah. So despite best planning and perfectly color-coded Gantt charts, translation can still fall short. So let's look at the gaps now. And I wonder that if you could give me real-life examples. For example, what was your biggest surprise or challenge when preparing your IND package? Or if you have done that. I know, Debra, you are not there yet. But
I think, yeah. So in fungal nail infection, there's a perception there are animal study models. There aren't any. There are models of athlete's foot because you inject trichophycin species between the toes. That's not what fungal nail infection is. And so we had to spend a lot of time discussing why we only had nail penetration data. And that was sufficient to allow us to demonstrate that it was worth testing. And we didn't have an animal model. We did no animal testing whatsoever. So again, it comes back to understanding your program. I think as AI builds up and maybe as things like organoids and we develop the data, we understand translatability more, then actually that's going to shift, because ultimately the agencies will be focused on safety. You know, a lot of the agency's view is efficacy is at sponsor's risk. You believe it's going to work. I just need to believe the patient's safe in testing. To your point about getting into clinic is actually not too difficult. But you need to have that confidence yourself that it's real reasons to believe. And your investors certainly are going to want to see that. So, yeah.
The biggest surprise I'd say I've had is having surprises. In that, you know, you put together your pre-IND package and, you know, you've worked on it for a period of time with a great team and you think, you know, this is foolproof. There's no way the regulator could possibly disagree with any of the wonderful arguments I've laid out here. I think about one example where we wanted to go into patients very early in phase 1. And there was a lot of good rationale for that. EMA said, yep, sure, that sounds great. FDA said, no, you need to generate healthy participant data first. So sometimes a little bit of window shopping can be good.
Yeah.
But I think probably the lesson I took from that is actually, if you want to do things that are slightly outside of maybe the norm, go to them before you put in your IND.
Yeah, definitely.
You know, go to them for a Type C meeting and say, you know, we are planning this. What do you think? And they will obviously give you a non-binding recommendation, but I think that can be really helpful to prevent the surprises. Because if you have a surprise at your pre-IND submission, that's going to impact your timelines.
Yeah, nobody wants to go on clinical hold. I think that's...
No, exactly.
Yeah, go on.
Oh, I'd say one of the big surprises as well. I recently come across, and that's a published data out there. So happy, that can be shared. So it was how the CAR T, the Novartis CAR T therapy got into the clinic. Well, actually, not just into the clinic; it got straight into patients for testing, and they only did mouse models. They could clearly prove and demonstrate that there's no human relevant animal model available. So they started this model that they built, a mathematical model for every aspect of how it would perform in humans would have its own equation, and all those equations were solved by a particular animal or in vitro model. So they had, from the mouse model point of view, they had immune-compromised mice. So that gives you an idea, but a humanized mouse, but it was immune-compromised. So that gave you an idea of efficacy because they also had tumors. And that data was used, but then independently and then conjoined in the model with a human immune model, which would tell you the immune reaction, but tells you nothing about the efficacy. And in the model, in the computer model, those two were conjoined to come up with basically the approval package for moving into patients. And that was, an innovative approach. It was the first time this was done. It was the first therapy. It was the first CAR T therapy ever that actually went into the clinic. But from an approach point of view, I thought that was absolutely fantastic. First of all, that they actually were brave enough to do it, and also the agency, agreed with it and gave them approval to go and do it. Now, I think we have to caveat that that's a super cutting edge therapy. It was in, I think, 2016 when they so it's already 10 years ago when they did that. So you're not going to get away with that for a small molecule. Your small molecule, you have to show good reasons why you don't do your two species in animals. Also with monoclonal antibodies, I'd argue, I think you need to have a strong argument on why you're not doing any animal testing. But there is definitely a path, and if you can put a good story together and the package and the data, I think, and be prepared also for the pushback, like what are they going to ask, and then be prepared to answer those questions. Ask them preemptively as well that, well, this is how I'm going to be doing it, this is what I'm going to be submitting. I think you've got a good chance to at least negotiate a path that you can follow.
I'm looking at the clock and we are almost at the coffee break. Okay, so I can ask maybe two questions. So I want to bring in the innovation and technologies. I'm super excited, and if you looked at my CV where I worked before, machine learning, pre-discovery, and all the sort of innovation happening there is amazing. And you look at the point when you have your candidate, you have your potent and selective molecule, and sort of valley of the death, or what's the best word? There's not a lot happening there. What would be, or what are you most excited or know about in that space and emerging technologies, innovation, that would make our life easier moving towards the clinic? Or it is all doom and gloom? And I'm going to ask another question, so it's not
I don't think it's doom and gloom. I think it's just different. As I said, that's the point where, yes, you have to address your preclinical, your translational toxicology and make sure first time in human testing is going to be appropriate and ethical. It's also where, as I said, you're moving into a phase where you're moving from laboratory to product. And you're starting to have to think about what those product elements are and how are you going to scale and how are you going to develop. And also that comes with how do I do my QP, my quality assessment, and get the qualified person release my drug product. And that's about having the analytics. So you end up in a point where you're not really working on developing your knowledge of the science anymore. You're developing your knowledge of your product. And it's a different phase, and that's really what pharmaceutical development's about. And I actually think for me, one of the things we've did is we bring that really early. So we use this thing called quality by design, which is all about understanding your quality of your product. And we employ those principles from day one in the lab. So that it means that then when you get into that translation, you can go to your manufacturer, you can say, make this, like this is how you test it, this is how you know it's good. These are the critical control factors. So you can try and abbreviate that and bring that in. And I'd love to think modelling has got a huge part to play in that, and I know process modelling has always been part of this, but I'm talking about trying to perhaps circumvent some of the more challenging bits of pharmaceutical development, stability, and all of those sort of things. It would be great if there were tools out there that would help us, because that would save a lot of money and time, actually.
I think there's also a space in that valley of the death where you've got your candidates, you've got your GLP tox data, you've got your animal PK, you're just trying to get everything together, as you've said, Dave, where you're starting to think about actually what do the details of the phase 1 study look like, the phase 2 and potentially beyond. And obviously there are a lot of companies out there looking at simulated clinical trials. And I think it's quite hard to simulate those things if you don't have any idea about the properties of your molecule. But at this stage, you've got a fairly good idea what your molecule is going to look like, with lots of caveats. So, now is the time to start those simulations. So actually, when you go into first time in human, the data you get out of that is much better quality to then get into phase 2 much easier.
Anything else? Or?
Got a question?
Oh, we have a question. Yes, Ron.
Yeah, I'd like to make thank you. I'd like to make a comment rather than a question to bring up a point. So I'm listening with great interest to the discussion, and there's one stepping stone across the valley of death that I'd like to bring up, and that's working, you're smiling because you know what I'm going to say probably, which is working with a rare disease as a first step. This is what my company takes as a strategy. And essentially, we find the patient associations in rare disease, and you realize that a lot of the problems that you brought up are already half solved by the time you build a good relation with them, because they bring extremely powerful disease models, including patient-derived organoids that get done very quickly because the community is extremely engaged. So you get brain organoids done in a matter of several months that come from real patients, with all the biobanking material. You get animal models that they've already created, and you get an extremely ambitious community that helps you through the lobbying with the regulators at early stages and are just lining up to be in your phase 1 as subjects. So I would just say it's a valid strategy to start, especially if you have a versatile platform, to start looking at a rare disease that could be relevant, and later, of course, you can expand it into additional indications and populations, which is far easier than doing the first step. So that was just a comment I wanted to make.
Maybe, I think that's an absolutely fantastic point, and it's become a business model in itself for assets or pharma company or biotech companies to go down that route, especially when we talk about modular aspects such as AAV capsids. You can buy them off the shelf and you can manufacture. Well, we understand the manufacturing a lot better now, and also the sort of the gene defect that's been solved. It's very relatively easy done. So you can take various different components and do it. Now, the key behind it is that there is actually a reward for for bringing a rare or ultra rare disease therapy onto the market in the US, and that's $150 million US, which you get as an accelerated approval pass Now, the accelerated approval process doesn't help you as a small biotech for ultra-rare disease, but you can auction it. And the last auction price was 150 million. So now, if you keep your prices down, and then the investor's calculation is, if you can get your drug, if you can get your development cost to 30 million with your rare disease or ultra-rare disease, obviously you need to be very careful about how you do your drug development. You can't spend too much money, but it's small trials usually. 20 subjects could get you through your clinical development. Now, if you keep your costs constrained and stick within the 30 million, you'll get an investor to invest that with the view that you can exit relatively quickly without even selling a single medication. You can give it away for free with that promise of getting that accelerated approval ticket, which you can auction for 150 million. So that in itself has become an own biotech industry business model in itself in the US.
Only for pediatric, and there have been
Yes.
only 68 of those granted so far.
Yes.
That's not guaranteed.
It's not guaranteed and it's not easy, but
The 2 years
yeah.
exclusivity for orphan you can get quite easily if it's an orphan indication.
I mean, I think the point you made is, you know, if you're working in the right thing, it's a good strategy. If everyone starts pursuing it, though, you start then chasing patients, which is the point I was making, that actually then impacts on your recruitment, because if everyone is going after the neglected and rare disease,
Exactly.
and there's only
The
a handful of
downside,
patients.
yeah.
So I think it is definitely a strategy, and it fits as long as it fits in your disease area. And again, it's just about trying to get early data, isn't it? Early evidence of efficacy, whether it's a biomarker, whether it's some early readout, is really part of the drive here for successful early development, actually.
On that note, shall we conclude? And we go for coffee. Thank you very much. I hope it was very useful for you.